Okay, welcome back to the Classics of Immunology Journal Club. And before we get started, I just want to remind you to like the video if you like it. I suppose you can dislike it if you don't want to if you don't like it. Click on the subscribe to this site because I make a new video at least once a week. Click on the little bell, and then you'll get notified when the new video appears. And then also scroll down and go to my website, and particularly for the Classics Journal Club, you can find all the classic papers on my website. If you click on the writing tab and then scroll down through there, you'll get to where we need to be. Today, we are going to review a paper from 1985, and this was a very important paper in the whole cytokine story. The authors were Bruce Beutler, Greenwald, Holmer, Chang, Pan, Matheson, Ulevich, and Tony Cerami. So the principal people were Bruce Beutler and Ulevich and Cerami. The title of this paper is Identity of Tumor Necrosis Factor and the Macrophage Secreted Factor Cachectin. It's published in Nature in August of 1985. Now, the background in this paper is that Bruce Beutler at this time was a postdoc with Tony Cerami at the Rockefeller University in New York City. And Bruce's background is very interesting in that his father was a scientist, and his father's name was Ernie Beutler, and he was a hematologist. And I used to be a hematologist, so I knew Ernie Beutler, and he was into glutathione and all kinds of things like that. Bruce, when he was growing up, was one of these curious kids particularly about nature. And when he got to be older, he thought he'd become a scientist like his dad. His dad told him that in order to prepare himself for science, he should go to medical school. So he went to the University of Chicago Medical School. And then his dad told him that he couldn't just stop there and go directly into science, because he'd be wasting most of his medical training if he didn't take the postgraduate years in the hospital, and because that's when you really learn to speak the language that was crammed into you in medical school. Medical school is really simply a language school. You learn a new language, and then you can talk to other doctors. But you have to be able to translate that language when you're talking to your patient. Anyway, so he did that. He went to the University of Texas in Dallas, Texas Southwestern in Dallas, did his house staff years, and then he got a postdoc job with Tony Cerami at the Rockefeller University in New York. So Tony was a biochemist, and so like his dad, they were in biochemistry and so forth. Tony had gone to Rockefeller University as a grad student, and he had gotten a fellowship when he was a young buck, and he went to Africa. And he got interested in this wasting that so many of the individuals had over there. I don't know exactly which country he was in, but a lot of the people had chronic infections. And when they had chronic infections, they just got skinnier and skinnier and skinnier. And this same thing happens to cancer patients when they have a chronic ongoing course for a long time. And nobody understood what this was all about. And so when Tony came back to New York and ultimately he set up his own lab, he decided that that's what he wanted to work on. There's a word, it's like the language school. There's a word for this wasting kind of syndrome. It's called cachexia. That's important here because there's a derivation of that is cachectin, which is a factor that made by macrophages that Tony found when he kind of started researching this. And in the introduction of this paper, they go into the fact that they had traced this cachexia that people were having to metabolic problems that these individuals would run into when they had chronic infections. The question was, why were they having metabolic problems, and what kinds of metabolic problems were they having? And one of the first things he did was when he got experimental animals and gave them chronic infections, and when he then drew blood from these animals, he noticed right away that they had a sort of a paradoxical situation where they had high levels of fat in their blood, serum lipids. So that was sort of a paradox. I mean, if they were skinny little animals, and then they had they couldn't seem to get the fat from the blood into the body, into the cells. They traced that further then, and they found that on the surface, particularly of fat cells, adipocytes, there was an enzyme called lipoprotein lipase, and that enzyme was very critical for clearing the serum of triglycerides. And triglycerides are the fat molecules that we come from the GI tract when we digest fat. So the fat comes into the body, and on the surface of the fat cell is this lipoprotein lipase. It cleaves the fatty acid side chains off of the glycerol, so you don't have triglycerides anymore. You can uptake those fatty acids into the fat cells, and then they are stored there. It turns out that they had found that the high blood lipids was primarily due to triglycerides, and that was due to a defective clearance of the triglycerides from the blood. They followed that further, and they found that then that was due to the fact that the lipoprotein lipase activity from cells from these animals was markedly depressed. And then the question was, why was that? And they found that if they took macrophages in cell cultures, and they were working primarily in the mouse, and they had a particular cell line that I'll get into later, that if they activated those macrophages with lipopolysaccharide, so-called endotoxin, that's in the cell wall of particularly gram-negative bacteria that is found in the colon. It's quite common that if you get infected with these bugs, that the lipopolysaccharide from the coating of the bacteria activates the macrophages. And then what they found was in the supernatants from cells stimulated by lipopolysaccharide, you would get a factor. And that factor then, when they were biochemists, so they started purifying that factor. They named that factor so that they could talk to each other about it as cachectin. Now, you might remember that, so this is 1980, in the 1980s, early 1980. From 1980 on is when Tony started doing these experiments, and this is 85, when Bruce was a postdoc with Tony. If you remember back to one of the other Journal Club articles that we looked into, was from Lloyd Old, from also in New York, across the street from the Rockefeller University. They had Memorial Sloan Kettering. He had identified by injecting lipopolysaccharide into mice itself. So you take LPS, you stick it into mice. They would then, in their serum, there was a factor that he called tumor necrosis factor. So this is where the story gets interesting. And of course, Rockefeller and Sloan Kettering, they were like the Hatfields and the McCoys. They were too close to one another. And so they were rivals rather than, friends. I guess you'd call them frenemies. So there the story was. And so back to Bruce and Tony, as they started purifying cachectin from these macrophage culture supernatants, they tested it for TNF activity. That the characteristic in vitro assay for TNF was a cytotoxicity assay. They used cells that were called L929 cells, which were mouse embryo fibroblasts. A cell line had been started in 1948, and it was one of the, everybody had L929 in their freezers. They simply repeated the TNF cytotoxicity assay that had been described had been utilized by Lloyd Old's group, and they found that their cachectin, even though when they got it really pretty pure, and so they got rid of any other molecules that might be in the supernatant, it had huge amounts of TNF activity, of the cytotoxic activity. So that set the stage for this paper, because they thought, well, maybe they're the same thing, or maybe they're very similar, and so we have to figure this out. And so that's what they set out to do in this particular paper. In the materials and methods, in addition to the fact that they made their cachectin by activating a cell line called RAW, it was a macrophage, mouse macrophage cell line with LPS, and to measure the cachectin, they worked up their own assay. They found a cell line that was very special, and it was also a mouse embryo fibroblast cell line called 3T3, but a special clone of 3T3, designated LLI. And this particular cell line looked like a fibroblast, mouse embryo fibroblast. But then if you gave it some hormones, if you gave it insulin and glucocorticoids, those cells would differentiate into fat cells, lo and behold. And then they had lipoprotein lipase activity on their cell membrane. And so Tony and Bruce worked up an assay that they could perform in microtiter plates, and so that they could do multiple dilutions of their cachectin and so forth. If they put cachectin onto these 3T3Li cells and then waited a while, and then they could measure the enzyme for lipoprotein lipase activity in an enzymatic assay, which is another biochemical assay. And so that was the assay for cachectin. And then I described the assay for TNF. So in the results of this paper, what they go through their purification method. And one of the reasons that I picked this particular paper is that this really shows the tour de force, what you needed to do in order to characterize any of these cytokines before you knew what was going on. You needed a biological assay which they got with the 3T3Li cell and the decreased lipoprotein lipase activity and so forth. And once you had that, you used your assay to then you would generate copious amounts of your factor, and then you would concentrate it. So they concentrated 50-fold in an Amicon filtration device, and then they said it was very, very important to add a non-ionic detergent to that concentrate that they had before they took it further, because otherwise they would lose it on primarily on the surfaces of all of the glassware and so forth that they were using at the time. So first thing they did was they put it and they took this concentrate and they put it on an anion exchange column. The anion is a negatively charged ion that migrates to the anode. So now we need to do some screen sharing. This is figure 1 here, and this is a figure of the data from their ion exchange column. So they would load onto the column their crude concentrate of this stuff, and then they would take fractions from this column as it dripped off from the bottom of the column. And what they did was they used a gradient of tris hydrochloride from 0 to half molar to 1 molar, and really the whole gradient that allowed them to elute off the cachectin activity, which is shown here in the gray peak here. They went from 0.3 to 0.5 molar tris, and that would give them their partially purified preparation. 60% of the protein just went right through the column and came off at the bottom, whereas the cachectin had been retained. They then they took 8, 9, 10, 11, 12. These are fractions, and they took the following edge of this peak, and they assayed them for protein by SDS page, and you can see here's the protein controls, and then here's fraction 8, 9, 10, 11, and 12, and this little arrow here depicts the what they thought was their cachectin. They then they then pooled these fractions that they got off of this column, and they went to the next step, which was, which I think is shown in figure 2, which was the gel filtration column that separates proteins on their molecular size. You can see the same sort of thing. There are all these fractions from this molecular sieve column. So if the molecules are small enough, they hang up in the end of the column, and then they will come off according to what their molecular weight is. And so this crosshatched bar right here are the fractions where they found cachectin-like activity. And here again, there is an arrow of the fractions that they found there. And so this is fraction 25, 26, and 27. They pooled that, and then they went on to, they took those and they pooled them together, and they went on to a high pressure liquid chromatography column, HPLC, that was separating the molecules according to their hydrophobicity. And they found that a very sharp peak of their activity eluting from the HPLC. And that then they took on to analyze it. And then you have to have pure protein. So one of the assays or tests for your pure protein is to do an amino acid composition, which they did. And they compared that to the reported amino acid composition of TNF, composition of TNF. And when you can see here, these are the percentages of the different amino acids in these two preparations. And you can see as you go down here, threonine, they have the exact same amount of threonine. Valine, same story. Alanine, same story. If you go down here, the histidine, same story. Arginine, same story. Cysteine, the same thing. So they thought to themselves, well, this stuff looks pretty similar to this is human TNF now, really. So it's mouse cachectin and human TNF, and that was very striking homology. So the next thing they did then was to take their purified protein and do a sequence of the amino acids from the N-terminus of the protein. And this is what they've depicted here. You can see that in the upper line, you've got this is the N-terminus here. This is cachectin. This is human TNF down here. And then this is the next iteration. So this is number 1 here, and this is number 19 here. So for the first 19 amino acids, you can see they've underlined where they have strict homology here. And so on the basis, these are the data. So we'll stop our screen share at this stage of the game and go back to go into the discussion of this paper now. So, you know, the name tumor necrosis factor is significant in this instance. And of course, Lloyd Old's group they were at Sloan Kettering and Memorial Hospital, and that's a cancer institute. And so they wanted to find something that had to do with cancer. So they named their factor that they found in the plasma of these mice, or the blood of these mice, tumor necrosis factor, because when they injected it into mice, if the mice had a visible tumor, you know, they would have just growing tumors that you could see with your naked eye, growing on their back where they were injected and so forth. And the tumor would undergo within 24 hours of the injection of TNF, they would undergo a massive necrosis, and it would just disappear within 24 hours. Well, my interpretation of that is that the reason that happened is that the TNF put these animals into shock. Their blood pressure fell to the bottom of the barrel. And because these were highly vascular tumors, they died of hypoxia because they had no blood supply. Anyway, that's important in this particular paper because Tony Cerami and Bruce Beutler had started with mice that didn't have tumors. They had infections, but so they wondered what the function of this TNF and or cachectin, if it was really the same thing, because that's what they thought, and that was the title of the paper. They had shown before they even did these experiments that there was a receptor on normal mouse cells and normal liver cell membrane that would bind their radiolabeled cachectin. So what's going on here? And they wondered whether or not the in vitro assay, where they would take tumor necrosis factor and put it onto tumor cell cultures, and the cells would die. They wondered what that was all about, and they speculated that maybe that's due to decreased protein synthesis in these cells, and that's resulting in the cytotoxicity. But this was the first paper, this paper that we're talking about right now, that indicated that this tumor necrosis factor stuff was not specific and particularly for killing tumor cells. There was a normal hormonal kind of an aspect to this tumor necrosis factor, and that stimulated them. There's a string of papers. If you look up Bruce Beutler and Tony Cerami after or before and after this paper, you find that what they found is that this was causing septic shock, essentially. That was what was causing the whole business, and that's why I say the hemorrhagic necrosis of there when Lloyd Old's group injected his mice, the tumors disappeared because they didn't have any oxygen. That's a pretty hard way to treat tumors, but exactly at this particular point in time, there was a group at Genentech in California that had cloned the gene for TNF and for lymphotoxin. And they started making large quantities of pure TNF, and the clinicians of the oncologists had jumped onto this because they'd started doing clinical trials and injecting it into patients. And there was about half a dozen of them all over the world that were competing to be the first to cure cancer with TNF. Well, what happened was that when they started doing dose finding safety studies with their TNF as they put it into human beings, and they found as they increased the dose, the blood pressure dropped. Not such a good thing. And so that was the end of TNF as a new anti-cancer therapy. And that was the beginning of the sort of the physiology and the immunology and physiology and understanding exactly what this TNF molecule was and how it played a role in basically the host defense mechanisms. And so the TNF became one of the triumvirate of the pro-inflammatory cytokines, interleukin-1 being another, and interleukin-6 being another, and we'll get into those as we go along. But those pro-inflammatory cytokines are what are released or responsible for the so-called cytokine storm that the COVID patients get into trouble with, and causing the leak in the capillaries of their lungs, so that all the blood plasma leaks from the blood into the lungs, filling up all the air spaces, and then you die. So there's a whole bunch more to these studies, to these stories, and we will get into them as time goes on. I guess the only other thing I want to say is that because the TNF and or cachectin, the way they produce their effects on the organism is by binding to specific receptors. So this is the paradigmatic thing for a hormone. That's what hormones do. Insulin, glucocorticoids, they all have specific receptors in their different cells in the body. And when the hormone binds to the receptor, it activates a biochemical process. And that is what we now call signal transduction. You change from a physical interaction of the ligand and the receptor into a biochemical activity. And therefore, that's how the whole thing works. So that's what cytokines are. That's why they're really, really important. And they can cause your death, but they can do good things too. So I'll stop there and just remind you to like the video and subscribe to the channel, and also click on the bell. And come back next week.